Bismillahirrahmanirrahim, my dear students of third year class. Today we will discuss the differences between the benign and malignant neoplasms. In the previous lecture we discussed the gross differences between these two, and today we will discuss the microscopic differences between the benign and the malignant neoplasms. So when we see a tumor, a neoplasm under the microscope, we find the cellular structures. These cells show different features in case of benign and malignant neoplasm. So first of all, when we see the individual cells, what are the differences? The cells of the benign tumor, their nuclear variation in size and shape is minimal. So they show minimum nuclear variation in size and shape. Whereas in case of a malignant neoplasm, the nuclear variation in size and shape, it is from minimal to marked and often variable. That is, you may find minimal change in shape and size, but in case of malignant neoplasm, many a times you will find marked differences between the shape and sizes of the nuclei in the malignant neoplasm. Then the cell division, usually benign, it shows diploid features. You will see the mitotic figures in benign rarely, and the mitotic figures that are seen in benign tumors, they show bipolarity, that is two poles are seen, whereas in case of a malignant neoplasm, the mitotic activity, it is multipolar. Multiple poles are seen. So bipolar, tripolar, tetrapolar. So multiple poles are seen. That we say atypical mitotic activity in case of malignant neoplasm, whereas typical mitotic activity in case of benign neoplasm. So in case of benign tumors, you will usually find low mitotic count, and high mitotic count is there in the malignant neoplasm. In benign neoplasm, there is always a retention of the specialization, whereas in case of malignant one, there is loss of specialization. That is, the cells that lose their functional abilities in case of malignant neoplasms when these are advanced tumors. So the structural differentiation, it is retained in benign tumors, whereas in case of malignant tumors, the structural differentiation, it shows wide range of changes. Now my dear students, you will ask, what is differentiation? So differentiation is basically the extent to which a new cell, it resembles its parent cell. That is the extent of resemblance to the precursor cells, the extent of resemblance to the parent cell. That is the differentiation. So in the benign neoplasm, you will find the structural resemblance of the new tumor cells with the parent cell, whereas in case of malignant neoplasm, the structural resemblance of the new cells is very less to the parent cell. It shows wide range of changes. Benign tumors, these are always organized, whereas the malignant neoplasms, they are not organized, they are disoriented. And the functional differentiation is usually seen, that is the benign tumors, they retain their capability of their specialization, their functional ability, whereas in case of malignant neoplasm, they lose their functional differentiation. Here the definitions: differentiation, the extent to which the parenchymal cells, they resemble comparable to normal cells. That is, their parent cells, how much these cells, they resemble to their parent cells, their normal cells. So this is called differentiation. Anaplasia. Anaplasia is loss of differentiation. that is when you can't recognize that which cells are these. That is, the cells, they do not resemble to their parent cell. Then we say that these cells are anaplastic cells. So what are the features based on which we say that it is anaplastic cell? The pleomorphism. Pleomorphism means the difference in shape and sizes of the cells. That the cells, they are different from each other, even on the basis of their shapes and their sizes. These cells, the anaplastic cells, they have abnormal nuclear morphology. That is, there is increased nuclear to cytoplasmic ratio. The ratio increases. In normal cases, the cells, or we say that in benign tumors or in normal cells, the cells, they have the ratio of 1:4. 1 for nucleus, 4 for cytoplasm, 1:4. Or you may find in some cases the ratio of 1:6 in the normal cells. But in case of malignant cells, in case of anaplastic cells, you may find this ratio as 1:1. Another feature of this anaplesia is hyperchromaticity. That is the feature of hyperchromatism. More darkly purple nuclei, that is hyperchromatic nuclei, are seen in case of anaplastic cells. And mitotic activity, it is abnormal, it is atypical mitotic activity that may show multipolarity. Dysplasia, it is loss of uniformity and architectural orientation of the cells. So this loss of uniformity is seen in case of pre-malignant conditions, when the cells are not malignant, when the tissue that we are observing, we say that these they do not show the malignant changes, but they are also not benign. So pre-malignant means the condition in between benign and malignant neoplasm. We say it is pre-malignant condition. Why we say pre-malignant condition? That the cells, they are showing the features of pleomorphism, they are showing the features of cellular atypia, they have shown the altered differentiation, that is their resemblance to their parent cells, to their normal tissue is reduced, and they are showing the features of atypia like hyperchromaticity, like increased nuclear to cytoplasmic ratio, like atypical mitotic activity, and cellular and nuclear differences in their shapes and sizes. So all these features are when observed, you will say the cells, they have shown the features of dysplasia with their loss of orientation, with their loss of uniformity. But yet you will not say that these cells, these tissues, they have shown the features of malignancy. You will not label that tissue as a malignant tumor. You will not label that. Why? When you are seeing all the features of dysplasia, loss of uniformity of the cells, you have seen the pleomorphism, you have seen the hyperchromaticity, the raised NC ratio, yet you are not saying that this is malignant. Why? Because to label a tumor as a malignant neoplasm, you must have seen the feature of invasion and later on metastasis. That if the tumor, it is not showing the features of invasion, and I have told in my first lecture that what is invasion? When the tumor, it breaks its basement membrane and it invades into the deeper, it infiltrates into the deeper tissues, into the deeper layers, we say that it is invasion. So if this important feature is not seen, is not recognized, you will say that this is dysplastic nature of the cell, but you will not label it as malignant until and unless you have seen the invasion or metastasis. So this feature, it can range from mild to severe. The sites, you may find this dysplasia in the cervix, in the bladder, in the stomach. Now we come to another important topic, that is in situ malignancy. What is this in situ malignancy? If you have, my dear students, clear your concept about dysplasia, you will very easily understand what is in situ malignancy. My dear students, when you find the dysplastic cells, these dysplastic cells are when found in the whole layers of epithelium. When these dysplastic cells, they cover whole layer of epithelium. There is no normal tissue at all. There is no normal cell at all. Say for example, I quote the example of squamous cells of the epithelium of our skin. We all know that our skin, it has the squamous epithelial cells that are present in the form of layers. So what are these layers? These layers are basal layer. From top to bottom, we will say the stratum corneum, then granulosum, then spinosum, then the basal. So these are the layers of cells, and these layers are present uniformly. But if that uniformity is lost and all the cells of all these layers, they show the features of dysplasia, yet no invasion is seen. That is, there is no breakup of the basement membrane, there is no infiltration of any cell into the deeper tissue, but whole of the epithelium, it is it has all the dysplastic cells. All the cells have lost their uniformity and have shown the features of atypia. Then you will say that this is an in situ malignancy. Epithelial neoplasms with features of malignancy, they have altered cell growth, they have cytologic atypia, altered differentiation, but no invasion through basement membrane. So such features are labeled as in situ malignancy. Here the picture shown to you. My dear students, this is the basement membrane, the basal layer. Above that layer, all the cells, they have their different shapes and sizes, they have disorientation, they have frequent mitotic activity, they have shown hyperchromatism, they have shown loss of polarity, but they do not invade the basement membrane and do not enter into the underlying tissue. So we say that this is the case of in situ malignancy. We call it as carcinoma in situ. Carcinoma in situ. In situ भी कहते हैं, in situ भी कहते हैं. Another feature, you can see these glandular structures in which the malignant, we should say atypical, in which the atypical cells, they have filled up the cavities, yet the basement membrane is not broken, and there is no infiltration of these malignant cells, these atypical cells. Let me say atypical cell. We will not call these as malignant cell. We will call these as atypical cell. So there is no invasion of these atypical cells across the basement membrane. So we call this condition as in situ carcinoma in situ. So thank you very much, my dear students. This is all about the carcinoma in situ. In the next lecture, inshallah, we will discuss about the nomenclature of